Hi, this is Dr. Ferguson. In this presentation, I'd like to introduce you to a relatively new class of drugs that's making its way to veterinary medicine, and that's the cannabinoids. These are not new from the standpoint of human medicine or from the standpoint of drugs that have been considered illicit drugs. I wanted to make sure that I gave credit to my colleagues at Canopy Animal Health, Dr. Minardi and Vaughn specifically, as well as my neighbor Bob Covington, who works for them. And they helped put together some of the aspects of these slides, and they're the company at this point in time, the Canadian-based company that is pursuing what I would call scientifically based approach to the use of some of these compounds in veterinary medicine. So as we alluded to, cannabidiol, which will be the major product that we're looking at for veterinary medicine, is a phytocannabinoid that's derived from the cannabis sativa plant, which we know as marijuana. Its current drug class is anticonvulsant, and that's the only approved use for this compound in the U.S. is for the treatment of seizures of a specific seizure syndrome in children over 2 years of age. However, of interest to us is the fact that, as we're studying, it's being used for pain and inflammation in investigational studies that are a major focus in veterinary medicine. It will also have there's also some interest, of course, in seizure and anxiety in domestic animal species as well, particularly the dog and the cat. Well, this busy slide is meant to basically allow you to study it offline. When you look at the PDF of this presentation, perhaps just those interested in the relatedness of the compounds that are produced by plants at the top. These are the phytocannabinoids. Then we have those that have been made synthetically. And then we have the endocannabinoids, and we always wondered, well, where do these products work? And these are the compounds that are considered to be endocannabinoids, are produced by the body, and up until fairly recently, until the cannabinoid receptors that we'll talk about were identified, and even still some of them are what are called orphan receptors, that is, clearly are receptors without a ligand. And so we're now fitting some of this picture together as some of these compounds are realized to act on these previously felt to be orphan receptors. Okay, so what is the endocannabinoid system? In our understanding of this system is that it's a series of receptors, ligands, and enzymes that modulate a variety of physiological processes. And the list of these processes includes memory and mood, those things we may have not be surprised by, but learning, pain sensation, of course, interesting to us for this unit, as well as appetite, not surprising to those that remember what THC does in marijuana. Now, there are three parts to the endocannabinoid system. There are the ligands that are the natural ligands, and an endamide is an example of that. I showed you the chemistries on the previous page, but basically these are lipid-based retrograde neurotransmitters. What does that mean? It means that they are acting at the postsynaptic neuron to influence the presynaptic neuron, and they bind to the cannabinoid receptors on the presynaptic neuron. The other parts of this are metabolic enzymes that degrade the endocannabinoids, and of course we'll spend some time talking about the function and locations of the endogenous receptors CB1 and CB2. Okay, this is probably also a slide that's pretty busy, but better studied perhaps offline. but I wanted to make some major points shown in text on the left as to what the receptors or cannabinoid receptors are doing with regards to the pain pathways. And for those of you that need a review of your pain pathway, this also would help you do that as well. Down here at the bottom right, you can see we have the cannabinoid 1 receptor. We'll try to point that out as we go through where they are. And then the cannabinoid 2 receptor. So it's difficult to see perhaps on this particular image. And then it's cannabinoid 2 receptor on glial cells. So we've known for some time that the cannabinoids can impact the emotional and cognitive manifestations, and obviously this is manifest through the CB1 receptors that are in the cortex, as well as some of the CB2 receptors that are on the glial cells. And in addition, and this is where it gets interesting with regards to pain, we have the impact of CB1 and again CB2 in the pathways that are normally associated with inhibition of ascending nociceptive transmission. This is not too dissimilar from how an opioid acts on pain management at altering the integrative part of taking the peripheral nervous system's input, which we get down here, and integrating it before it is perceived by the cortex. And so we have obviously both types of receptors involved in that. We also have activation of descending inhibitory pathways that are associated with essentially both types of receptors in the medulla. And we have the effect of both receptors in the dorsal root ganglion that can lead to altered neuronal sensitization. And finally, we have both impacting the nociceptive transmission in the sensory neurons. So basically the potential for cannabinoids to impact pain is at almost every level of the pain pathway, and that leads to these compounds having great potential with regards to pain management. Well, I'd be remiss if I didn't at least try to present a little bit about how these compounds work and how where these receptors are located, and why cannabinoids not only are impacting potentially pain but also inflammation. And on the left is all of this in text, and I'll just try to carry you through it briefly, but it's another slide that you might want to study offline. First of all, it's known that basically cannabidiol acts as a negative allosteric modulator on the CB1 receptor. What does that mean? It means that it doesn't bind by itself directly to the receptor, in at least not at concentrations that you can achieve pharmaceutically, but it seems to work allosteric to it, meaning at a site that will impact the function of that receptor. It's also an agonist for what's called the transient receptor potential ligand-gated cation channel, which is shown over here, the cation in this case being calcium. And as an agonist, it's going to modulate that function as well. It's not shown on this particular image of a neuron, but it felt to be CBD is an antagonist at the tumor necrosis factor alpha receptor, and this is going to be associated with its anti-inflammatory activity as well as its pain modulation effect. It's an agonist at the serotonin receptor, which has a lot to do with perception of good feeling, etc. enhances the adenosine receptor signaling through the A2A receptor. And here's where we get into some of the orphan receptors that are now finally finding their ligands. One called GPR55 as well is now becoming more and more studied. That's this one here. And it seems to work through various G protein coupled mechanisms to impact and increase calcium intracellularly. You can imagine how this would impact the function of a neuron that is presynaptic. And then you have the GPR18, and while it's shown here, what we call the neuron, it's probably best expressed in leukocytes, and it seems to have its function as an anti-inflammatory function. So basically, CBD cannabidiol is felt to be non-intoxicating. What does that mean? Its major effects are not those that we associate with the altered emotion, etc. So at least not in the CB1 receptor. These are not likely to be leading to this kinds of side effects that we normally are considered the reasons why marijuana, for example, is considered a therapeutic / recreational drug. Okay, so continuing on with how CBD is involved in pain perception and transmission, basically we know what we were talking about is that they impact membrane potential and signal transduction. And so what the net effect is depolarization and rapid desensitization. Through the adenosine receptor, basically they alter the transporter for adenosine, and this has an impact on cellular function as well. And then they impact pre- and postsynaptic G protein coupled receptors and impact calcium, etc. And they interact with serotonin receptors, the GCPR55, the PPAR types of receptors and voltage-gated cation channels. That was the TRPV receptor, all of which modulate nociception or transduction of painful signals. And so how does one alter the endocannabinoid physiology? In this case, it's through inhibiting reuptake of anandamide, which is an endocannabinoid, and altering its hydrolysis. And basically, anandamide is felt to modulate pain through its effect on the CB1 receptor. So far we haven't really spoken too much about what we know about them in dogs and cats, and specifically the work of the people at Canopy Animal Health are currently looking at some of the potential adverse effects one might see with regards to CBD in oil products. And so they did a study that the adverse effects, that's AE here, were similar in number and type to placebo. So here's the placebo, and they had a lot of GI side effects. And here's the CBD in oil shown here. And why they were GI side effects, they could only postulate the carrier oil volume, or oil gavage led to some emesis, nausea, and diarrhea. But the point is, that's why you do control. They could not distinguish any other obvious negative effects, adverse effects associated with the administration of these drugs. And obviously they were going through the first phase of proving safety of what was they feel to be a therapeutic dosage, concentration, and that they say basically showing through this study that the adverse effects are minimal. So with this general background, we wanted you to be familiar with the endocannabinoids and particularly the cannabidiol product that is being developed for the dog. This is just one of many of the compounds that are found in the phytocannabinoids, and yet it was known for some time that outside of CBD itself, the others may have more toxic side effects than the CBD itself. So it's very important to be using pharmaceutically prepared forms of CBD and not, for example, hemp oil or marijuana itself in a dog or cat, because this can be much more toxic because of the other compounds that are in those plant products.